I'm Dror Sapir. This is luminescence. Luminescence is a phenomenon in which materials emit light as a result of stimulation. This lab is about exploring properties of photoluminescence, electroluminescence, chemiluminescence in order to better understand luminescent sources and ways to stimulate their emission. A few goals is measuring the absorbance and emission of different fluorescent and phosphorescent compounds, including dyes, minerals, glow sticks, glow stars, and miscellaneous objects. Illuminating fluorescent compounds with different sources to observe which wavelengths stimulate emission of each. Measuring fluorescence lifetimes of different objects and compounds. Determining minimum concentration of fluorescein required for fluorescence to be detectable, and initiating chemiluminescent processes and
I'm
measuring the
starting
emitted
this lab
spectrum of each
having
of these
the white
stimuli.
light source in the cuvette with water in the holder. I'm using the ocean optics setup, and here is the spectrum of water. Next, I'm also going to put these other cuvettes with different materials, and those include dilute rosin Y, dilute rhodamine B, and quinine. So now that I have quinine in there, that completes all of the compounds I'm going to do. And now I can analyze this data.
Here are the results for the emission spectra of fluorescent dyes. We have each dye color coded differently here, and we can see that the ones that stick out the most are the rhodamine B and the eosine Y. Both of those have a peak in the visible range and a peak in the IR range. And the shapes of the peaks are very similar, and that made me think that there might be similarities between the molecules that should be looked at. So rhodamine B is on the top and eosine Y is on the bottom. There are some similarities you can see right away, like the carbons, but the edges of the molecules are different. But what I think is important to note is the core of the molecule, where that oxygen is on the bottom of eosine Y, on the top of rhodamine B, and really that structure around that oxygen is almost the same. And if one of the molecules is flipped, then they really look very similar. And I think that does have to do with why the fluorescence is so similar.
So now at this point, I'm going to take each one of the dyes separately, and I'm going to illuminate it here with the different light sources to see what's going to cause more fluorescence. And my hypothesis is that anything closer to the UV or to the violet end of the spectrum is going to cause more fluorescence than anything closer to the red part of the spectrum.
Just a little bit of background for why I hypothesized what I did. In this picture, the violet arrow shows, say, 405 nanometer light. It raises an electron from the ground state to, say, 3 at S1. And now that electron is going to have some non-radiative transitions, which are going to cause it to lose some energy. And from that point, when it wants to go back to ground state, the gap is going to be smaller, and that gap is going to represent longer wavelength light with lower energy. And that's why that light is going to be more green or more red than that original violet.
So this is the eosine Y, and I'm going to illuminate it with the red laser, and what we can see is a lot of scattering, but this is the same wavelength that's going in, so this is not fluorescence. And here we go with the green, and I believe this is fluorescing. It's fluorescing yellow, and the violet's of course going to cause stronger fluorescence. Same thing with quinine. The green does not cause it to fluoresce, and if that's true, then the red is also not going to cause it to fluoresce. See that right now. There we go. And this is gonna, it fluoresces blue, even though the 4 or 5 nanometer violet kind of overtakes it when I'm trying to film it, but I can see it with my eyes that there's a wavelength difference.
A little about the results. This table shows, as expected, that the 405 nanometer LED caused stimulation in all of them. The red laser, the long wavelength, that was not enough energy to stimulate emission in anything. And as far as the green laser, it didn't for the most part, except it did stimulate eosine, which gave a yellow emission. And lastly, the blue laser was actually 405 nanometer like the LED, so that was not valuable data. So a little bit of background knowledge about me. I operate a YouTube channel that revolves around optics and science, and within the scope of that channel, I did an experiment where I took a glass and filled it with tonic water and ran 3 lasers through it. So what I wanted to show in this image is that the red and the green clearly are not fluorescing, and you can tell that that's the same wavelength going in, and the 405 nanometer is. Here are a couple more pictures that I took, and what I wanted to show is both in the left and in the right image, the wavelength difference between the incoming laser and the fluorescence is very clear to the naked eye. So on the left it's green and on the right it's cyan. So why is this important? Fluorescent and phosphorescent materials can be used for a wide range of applications. Biomedical research often requires one type of cell to be identified in a larger system. A good method for identifying such a cell is to dye it with luminescent dye. In a larger system, many cells may need to be identified, which would require dyes that emit a wide range of wavelengths. Understanding the chemical and optical properties of fluorescent dyes sets the stage for discovery and invention of new dyes, as well as more efficient sources of stimulation. In conclusion, the field of luminescence has been fascinating to me for a long time. I believe understanding the processes behind it is a vital part of understanding optics. This lab allowed me to explore a wide range of luminescent processes and the sources that initiate them. I believe this knowledge could be put to use and further expanded when researching fluorescent dyes for specific engineering applications in the future. Thank you.